So today I will be talking about bioconjugation because it is this year's theme for biotech. Let's get started. Let's start with what bioconjugation is. So bioconjugation is basically biotech's chemical superglue. It covalently links biomolecules with synthetic payloads like cancer drugs, fluorescent dyes, and nanoparticles. Antibody and cancer drugs become a tumor-seeking ADC, and protein dyes light up disease markers for imaging, but it must happen in water at body temperature to keep proteins alive. Traditional methods randomly hit 20 to 80 sites per protein, which creates disaster. This randomness creates a health crisis. We can see here 84% adverse events, and we have 29% grade 3 to 4 severe, and 22.8% liver toxicity. And as for neuropathy, that's 25%. And we have even more health risks down here. Let's move on to societal impact. Here we have poor and rural patients losing access to complex therapies, which is horrible. As for economics, we have $50 million in annual manufacturing losses, and that is a huge hit. We have 70% FDA rejections, and we have hospitalizations, which is $100,000 per patient for severe AEs. And that is lost productivity right down. Let's look at the problem as a whole. So, traditional bioconjugation lacks precision. Lysine chemistry modifies 20 to 80% sites randomly, so DAR mixtures. Some antibodies overdose while others do nothing. Cysteine methods cause dimerization, and there's 40% activity loss. Let's look at our solution now for that problem. We will have unnatural amino acid code expansion. Here, we're going to hijack protein synthesis using an engineered tRNA pair that inserts this really long word, d-acetophenylalanine, at any chosen position via a TAG codon. Now, every protein gets exactly one handle where we want it. Click on drugs via SPAAC chemistry. So that would be DAR is equal to 2, perfect versus traditional DAR mess. For our results, we're going to have 95% batch success, 4 steps per 2 days versus 7 steps per 5 days, which is way better than before. And then we're going to have 5 to 10 times the therapeutic index. So that is really good. Now, how can we apply that solution in real life? Well, we already see this at Genentech with the UUA expansion technology. Now, Genentech HER2 breast cancer, it is engineered trastuzumab with PAZF away from binding sites using CHO cells, SPAAC plus MMAE drug, and that's exact DAR is equal to 2. That's a lot, but we have a mouse model now, so 4 times tumor regression, 70% complete response versus 20% lysine ADCs. And then we have 60% less liver toxicity, and it meets FDA homogeneity standards, which, it also accelerates clinical trials. So they're doing a very good job. Moving on, we got Scripps research. So Scripps Alzheimer's imaging they added p-acetylphenylalanine to amyloid peptides. Oxime ligation with NRI fluoride force post-injection. What are the results? We have 100% site-specific tagging, 48 hours earlier plaque detection than PET, and 3 times signal to noise. Scaled from 10 milligrams to 500 milligram batches without yield loss. Positions tech for human brain imaging. So we know that Scripps Research Institution, they apply our solution really well for human brain imaging. Thank you so much for listening to this presentation. I hope you learned at least something from this presentation, as we did our best job on this. Have a good day.